I actually got my interest in longevity as a first-year graduate student. I went to graduate school thinking I was going to do X-ray crystallography or structural biology or something like that. And I heard a talk by Lenny Guarente. This was at MIT, about how his lab, just in the last few years, this was 1997, had started studying the genetics of aging, and got turned on by the idea, and joined Lenny's lab, did my graduate work studying aging in yeast with Lenny, worked on SIR2, which at that time, the word sirtuins didn't exist. SIR2 in yeast is the founding member of the sirtuin family. So it was really my PhD thesis work that established SIR2 as a longevity factor, and sort of led to a lot of interest in sirtuins in the aging field. Spent about a year and a half right out of graduate school at a startup biotech company. We were a couple of decades early, but our goal was really to develop what at that time we were calling biomarkers of human aging. Now people call them biological aging clocks. The technology wasn't quite ready yet, but it was a super interesting experience. Then after about a year and a half doing that, did my postdoc with Stan Fields, started working closely with Brian Kennedy at that point, again, studying the genetics of aging. Brian and I are fantastic friends. He's one of my best friends in the world. Had a super productive collaboration with him. We've probably published more than 100 papers on different aspects of aging biology. Started my own lab at UW, University of Washington, in 2006. Spent about 18 years there, really interested in understanding the conserved mechanisms of aging that are shared across the animal kingdom. About 10 years ago, started something called the Dog Aging Project, very much in that same vein of trying to move from what we'd learned in laboratory animals into the real world in companion dogs. And then about 2 years ago, left my academic primary position to start a company called OptiSpan, which is really at the very far end of the spectrum of trying to bring as much as we can in the realm of geroscience and longevity and health optimization to as many people as possible. So I would describe OptiSpan as a healthcare technology company, although we do have a clinical program where we are seeing patients and working with them to try to optimize their health as much as possible. Along the way, spun Aura Biomedical out of my lab. And so we developed technology there, really aimed at trying to attack a problem that I first started picking up on about 10 years ago, maybe a little bit more than that in the field. I feel very much like the field narrowed too quickly, became far too entrenched in the hallmarks of aging paradigm, and really largely, and this isn't true of everybody, but largely stopped looking outside of the hallmarks of aging. And as a result, we know a lot about the hallmarks of aging, and we know a lot about interventions, drugs that target the hallmarks of aging. We know almost nothing about what else is out there. And so I really set out to try to tackle that by working to develop technology that would allow us to screen longevity interventions at scale. So that was really the rationale behind the WormBot. So we created this system that we believe will allow us to test more than 1 million interventions to establish which of them have much larger effects on longevity than anything we know about today. And that's really the goal of Aura Biomedical, is to take that technology and bring it to the next level.
Amazing, super comprehensive. Really appreciate all of that. Super cool story, honestly, and really love the way you've kind of tried to break the entire paradigm in thinking and resource allocation in longevity outside of where it's hyper focused and potentially overfit to the existing hallmarks that people know about. So really appreciate you pushing that kind of thinking. That kind of out of the box thinking is something we try to aspire to as well here. But I mean, like, I think you explained kind of the rationale behind the WormBot. You know, you want to screen all of these compounds at scale. You think that, C. elegans is a good model organism to test these compounds in. I guess maybe let's start with that, like building up the argument for the WormBot. Like, why is C. elegans such a strong disease model or model organism?
Sure. Yeah. And so I would even take a step a step back from there and start from, again, this idea. I mean, you can ask the question, first of all, is my thesis that we narrowed too quickly, is that valid? I mean, I think you could make an argument one way or the other. If you look at the number of interventions that have been tested for their effect on lifespan, that's a good place to start. And we don't know the answer to that. We don't know the sum total of that in all of human history, but we can look at the largest publicly available database, which is called Drug Age. Anybody listening to this can look up Drug Age on Google and go see this database. I don't know how many interventions are in there now, but it's on the order of 1,500. That is the sum total of all the drugs in all of the literature that have ever been tested quantitatively for their effect on lifespan. To me, that seems pathetically small if you think about all the interventions that are possibly out there. So I don't think you can argue with the fact that we have done a really, really poor job as a field of exploring all the possible things that might affect lifespan. And that's just looking at single drugs, let alone double, triple, quadruple combinations. And if any of you are in the biohacker space, you'll know that many people are taking 20, 30, 100 different things. So we haven't studied that at all for lifespan. And so if your goal is to say, okay, we need to explore intervention space in greater detail than 1,500 drugs across all animals we could possibly test these things in, you have to ask yourself, if you want to do this at scale, and even there, if you think about all of intervention space, double, triple combinations, 1 million is only a tiny fraction, but you got to start somewhere. So if you say, okay, let's think about how could we test 1 million interventions, and I'm using intervention as a catch-all for 1 drug, 2 drugs, 3 drugs, 10 drugs, gene therapy, other environmental changes, just a catch-all term for ways you could modify the environment to affect lifespan. If you want to test 1 million, you have to start somewhere. So you ask yourself, what animal could we test 1 million interventions in? Well, people aren't going to work for longevity, obviously. Dogs aren't going to work. Mice, maybe, but that would be 50 million, 500 million mice you'd need to do that experiment, and they live 3 years. That's probably not realistic. So you get to animals that you could actually potentially develop the technology to do this at scale with. And we settled on C. elegans for a variety of reasons. I think they're relatively short-lived. About 3 weeks under standard laboratory conditions, we can know whether a drug is increasing lifespan or not. You can grow billions of these animals at scale in the laboratory. And we thought we could potentially automate the lifespan analysis using digital imaging and artificial intelligence and robotics. And so that's why we set out to use C. elegans. Now, will everything that works to increase lifespan in C. elegans work in a mammal or in a human? No, of course not. Some things will increase lifespan dramatically in C. elegans, and they'll be toxic in mice or toxic in people. But I think we've learned that the underlying biology of aging is conserved enough that if a drug robustly slows biological aging in a worm, there's a reasonably good chance it's also going to slow biological aging in a mouse. And I would argue a reasonably good chance it's also going to slow biological aging in people. And we know of multiple examples that fit this paradigm of being shared from worms to flies to mice, probably to people, although again, we haven't definitively shown we can increase lifespan across the population of people with any intervention, maybe with the exception of exercise and healthy diet. So it seems plausible that this is a good place to start. Many of the things we find in worms will work the same way in mice, probably the same way in people. So that was sort of the rationale for why we settled on C. elegans. We worked for a few years to create the robotics and the digital imaging and train the neural network to get a sort of version 1 WormBot that we could test thousands of interventions on. And then tried to get funding through the academic world. I won't go into all the problems with academia, but I will say it's really hard to get funding for projects that are what people call a fishing expedition. In other words, they're not mechanistic. We're not trying to say we want to find the next mTOR inhibitor or the next AMP kinase activator. We're saying we want to look as broadly as we possibly can and find out what works. But really, NIH reviewers want you to focus on detailed mechanism, not find stuff that works. Sorry, that was a little that was a little facetious, but also partly true. So we couldn't get funding to do that sort of a large screening effort on the academic side. decided to spin the company out and see if we could get traction on the for-profit side, created Aura Biomedical, and are working diligently to find the next generation of best-in-class longevity interventions.
Awesome. I guess so. All super interesting. I think there's so many different directions we could take the conversation in, but I think, frankly, the one that's most relevant to the listeners here will be around the funding side. So I know you said you didn't want to get into the issues of academia, and I know that Aura has kind of taken a different approach, going for VC funding and trying to raise through private capital markets for the fishing expedition. But maybe if you wouldn't mind just briefly going through the issues with both. If you don't mind, like the two main options are, grants from, the governments,
Sure.
Predominantly the NIH, and then with, private companies, you're talking about VCs, maybe some grants as well. But would you mind just like kind of describing what that process is like? Is it a good process? Do you think they get it? Do you think they're missing things? Are they incentivized the right way, et cetera?
Yeah. So I do think the incentive structures are misaligned for at least this kind of a project, this sort of large discovery. Let's figure out what we don't know. Let's find things that work way better than the best in class right now. I think neither of those, the current crop of nonprofit funding opportunities or the traditional sort of for-profit venture capital approach are aligned with that sort of a mission statement, so to speak. So on the for-profit side, you can obviously recognize that if you have, let's just say you've got 10 drugs that work way better than anything we currently know about for longevity, starting in worms. You validate that in mice. Once you're confident they work robustly in mice, then you have the opportunity to move into a traditional drug development pipeline. So I think that would be the model that you would most easily envision from a venture capital perspective. But this is way earlier than most VC are used to investing in this sort of an endeavor. And I mean, for better or worse, most venture capitalists are cookie cutters. They want to do what everybody else is doing. They want to follow the pack. They're going to fund a traditional, one drug, one target kind of biotech development project. So it's not what they're used to seeing. It's challenging to get funding for this kind of a project in that environment. Not to say there aren't other paths that Aura can go down and is exploring, but for this particular mission, I think it's ill-suited. And the same thing's true on the academic side. Again, it's not a typical sort of very mechanistic project. For big science, the kinds of things that typically get funded nowadays are either directly in humans or kind of multi-omic sorts of approaches. That's just what's popular. So it's not well fit for those sorts of funding mechanisms either. I do believe there are funding bodies out there who can recognize the potential impact on the field and could potentially fund a project like this at the scale that I would like to see it funded at. But that hasn't happened yet. And again, even though I like to think of myself, I say often sort of half jokingly, I'm a recovering academic, I'm still very much of an academic mindset that I would love to have the resources to create a large open access database of 100,000, 1 million, 2 million interventions, give it to the community, and then let people take that and learn a lot about the biology of aging from it. And I'll just give you one really obvious use case. So there are a lot of people who have published papers or who talk a lot about how artificial intelligence is going to solve aging. I'm not that optimistic that AI is going to solve aging, because when your training set is garbage, guess what you're going to get out? If you put rapamycin and metformin into your training set, guess what you're going to get out? What we really need is a much larger training set so that these AI tools can actually live up to the potential that they have to really do novel discovery and put together patterns. And I don't think our training set is sufficient at this point to do that. So I think if we had a 1 million intervention database, that's a huge training set compared to 1,500 now, where I think you could actually start to achieve some of the promise that these new AI tools can bring to the field. So that's just one example of how having a very, very large open access database, I think could be sort of a sea change moment in the field of moving us into a new era where at least we'll start to understand, what we know today, is it 80% of biological aging? Is it 0.8%? I don't know. My guess is it's closer to 0.8%. We'd start to learn that if we could have this very large data set to query.
Makes a ton of sense. I think we're certainly aligned that, on the current path we're on, there's no chance AI gets the necessary training set to, start to be able to predict what interventions should we be taking on a large scale. It's not even a question of quality of training data. It's just lack
Yeah.
of training data.
Yeah.
We would love for the data to be messy, as long as that there's enough for us to parse through and sift through. And, with the way that these compounds get tested and the data gets stored, it's all sequestered within all these individual biotechs. There is no way to kind of gather all of this data into a single database where you can start to build, machine learning models that have sufficient training to do anything predictive. So I think we're totally aligned there. I think where we hope to kind of provide this gasoline needed to fund this massive throughput of compounds is via crypto. So this kind of like fishing expedition is something that many traders in the crypto space do every day. I mean, we called this space from doggy coins to doggy years. There's about 100,000 doggy coins that get launched per day. Everyone trying to figure out which is the high quality one, right? And so one could analogize this very much to high throughput screening of compounds, trying to find that one gem, right, out of this sea of garbage, right? Just to be able to filter through the signal and noise. We find that markets are super efficient at being able to sift the signal from the noise, given enough time and training, et cetera. So we think it's a good application to your exact problem that you're kind of highlighting. But yeah, really appreciate you highlighting that. But I hope that kind of makes sense, which is, we want to provide that speculative energy to the fishing experiment that is, sifting through all these compounds to see which ones have a high signal. Cool. So yeah, really appreciate you kind of getting into this with us. I guess one question I'd have for you is like, from an outsider's perspective, just looking in at the crypto space, what is your personal experience with crypto, if any? And, do you think there's a chance that we're on the right track here, that, actually letting individuals fund these individual compounds directly may be the, most scalable system, let's say, to screen these compounds and get them funded?
Sure. So I think from a sort of higher level perspective, I certainly have seen that members of the crypto community are very enthusiastic about longevity and have been some of the most ardent supporters of longevity science. In fact, the expansion of our clinical trial in dogs as part of the Dog Aging Project was funded in large part by members of the crypto community, including a large donation from Brian Armstrong, the CEO of Coinbase. So I know there's a lot of cross pollination and cross interest there. I will say I personally don't have a deep enough understanding of the inner workings of crypto to be able to, I feel, confidently comment on the opportunities for this kind of a model, right? I love the idea where people could speculate on interventions and somehow be rewarded for how that intervention turned out. I just don't have enough of a understanding of the inner workings of how you actually make that happen. So I think it's a really cool idea. And if there's a way to do it where, everybody feels confident that it's being done properly and people can participate in the scientific process and potentially do well financially at the same time, that seems like a win-win to me.
That's exactly the system we're trying to create. And, not to go into it too deeply, because that would probably take too much time than we have. What we're doing on the pump science side and within the decentralized science space is trying to tokenize the IP. So basically each compound, whatever IP it generates, whether it's a patentable compound or the IP is just the data that's generated, the token holders have the right to license. So that's kind of the the underlying structure behind the token that we think gives it value and enables this kind of whole speculative system. So that's kind of how we're doing it. And basically, like, anyone can have an idea for an intervention that they think may be promising. If other people think that idea is promising, then they can also, fund that experiment, so to speak. And then if anything comes of value, then that IP can be licensed and reward the token holders. So that's kind of the basis for what we're trying to do. And we agree with you that it starts with WormBot because you have the most scalable instrument to develop the largest data set that is high signal, low cost, and fast turnaround time of probably any other experiment out there. Maybe a follow-on question would be, what next? Like, let's say a compound extends life significantly in the C. elegans. It's been run in triplicate, so we have good reproducibility. What should we do after that? And what do you kind of see as this whole pipeline? Let's assume, if you don't mind, that we don't have to get VC funding to do any sort of like disease-based drug discovery. We can just develop it to extend lifespan.
Sure.
How would you de-risk those compounds?
Yep.
Or interventions?
Yeah. So, I mean, I think obviously it's going to depend a little bit on the nature of the compound or the intervention. So, the nice thing about the platform is it's really agnostic to what the intervention is. And what I mean by that is it could be a natural product, it could be a pharmaceutical, it could be a gene therapy. You just have to be able to deliver the intervention to the animals. And then the platform will measure the effect on lifespan. And I haven't talked about it, but can also measure the effect on multiple health span metrics. So it really doesn't matter what the intervention is, but the way you would then de-risk that intervention or develop it further would depend on the type of intervention that it is. So, for example, if it's a natural product that is generally recognized as safe, there's really no barrier to people taking that product right away and to bringing that to market. In fact, there's a good chance people will already be taking it. When you're getting to combinations of things, I think that's where it gets interesting. If you have a combination of natural products and you get synergistic effects on longevity, then I think that's really interesting to think about from a validation perspective. How do you validate it outside of worms? So there are a few options. You could go to another invertebrate like fruit flies. I think there's some value there. To me, though, I think the next really interesting question is, does it have a similar effect on longevity and health span in a mammal? I would approach that by testing the intervention in aged mice. And this would be independent of whether it's a natural product, a pharmaceutical, a gene therapy. I think to really have confidence that you want to move this forward into either companion animals or humans, I'd like to know that it has a similar effect in mice. The nice thing is now aged mice are commercially available. So you don't have to wait 2 years to get them to the point where you can do the interesting experiment. You can buy aged mice. You can then test whether or not the intervention in the context of aged mice improves multiple functional measures of health span. You can do that in a few months. So several people are doing this. Brian Kennedy's lab at NUS, National University, Singapore, has sort of become experts in these kinds of short-term studies of health span in aged mice. So it's not a super long-term experiment at that point, once you know what to test to get a readout. Realistically, 6 months, you can, I think, be pretty confident whether or not your intervention is targeting the biology of aging in aged mice. Ultimately, you'd want to know what the effect on lifespan is. But I think if you see a really robust positive benefit on health span metrics across multiple tissues and organs in 6 months in aged mice, you're starting to feel really good about the potential of moving this forward towards a marketable product. So again, this is when it starts to depend on what the nature of the intervention is. If it's a natural product, in principle, you could go directly to market. Again, personally, I like to know whether things work. So I would hope that that gets paired with some clinical studies to actually demonstrate efficacy, whether it's in companion animals or in humans. If it's a pharmaceutical, then you would be looking at sort of the more traditional drug development pathway, potentially getting an IND from FDA, going through safety trials, things like that. Same thing if it's a gene therapy or some other, more exotic type of intervention that you could imagine. But again, very generally, I think validate in aged mice, And then if you get a really strong signal there, start to think about the path forward towards further development or going to market.
Super useful. So just to kind of say this back to you, the C. elegans is kind of the entry point to the de-risking pipeline. Then you'd recommend, flies, potentially other model organisms, but very, very likely flies. Following that, it would be a health span and then lifespan experiment in aged mice. And health span, I imagine, I don't want to put words in your mouth, would be dissecting, looking at the organs, looking at the pathology of those organs to see, does it have an organ-specific anti-aging effect? Is that
Yeah,
is that a good recap?
although I'd give a couple of tweaks to that. One is, I think if you have a good system for going to flies, do it. I don't view that as a necessary step. And then in mice, I would say, yeah, you probably want to look at molecular markers, tissue pathology, quote unquote biological aging clocks. We now have epigenetic, metabolomic, transcriptomic, proteomic clocks, but also function. There are really good ways to assess frailty and function in mice. Well, I don't know if I should say really good, but there are good ways. There are ways that are quantitative that people are working with that are getting better. All the way from frailty indices to ultrasound to look at the heart, for example, to cognitive assessment. So I think you'd want to look at functional measures as well to get as comprehensive as you can an assessment of sort of health across multiple tissues and organs. But yeah, from a general sense, that's the idea, is you would try to do that in a 6-month period in aged mice. And we now know that multiple interventions can improve trajectories in already aged animals. So that's a nice place to start, where we already know that we can see effects. The last thing I would add, though, is it's my expectation, and even the data that I've seen from the data that Aura currently has in-house, which is a tiny fraction of the million that we're going for, the effect sizes we're looking for are much bigger than what we already know about, right? So again, I think the opportunity is we're not just looking for things that have positive effects in mice, we're looking for things that have big positive effects. And so that's what I would really be hoping to see before I get too excited about moving things forward. Again, maybe I'm setting the bar too high, but I don't think we need another rapamycin. We want something that's way better than rapamycin. So that's what I'd really be looking for at that mouse stage, is something that's more effective than our current best in class. And this is a little bit of a tangent, but I think people in the field really should be asking, why is it that nobody's finding stuff better than rapamycin? And I think it's because everybody's looking under the lamppost at the same network, which is the hallmarks of aging, and nobody's looking outside of that. I know that it is possible to find things that are better than rapamycin. Aura's already done it. If we look at 1 million, we're going to find a lot of things that are way better than rapamycin. That's what we should be going after.
100%. And one person's ignorance is another person's opportunity. So here we are, ready to dig through the weeds with you. Yeah, no, I'd love to press on this point, though, only because it's of supreme importance to us, which is this whole pipeline, because frankly, it's a pipeline that we're trying to turn into a protocol and build an entire funding layer around, right? So, where Aura is the entry point and flies are kind of this second step. This third step we really need to get right, if we are to scale, crypto funded longevity experiments. And just to be completely transparent with you, we are totally thinking about aged mice as well. In fact, we're about to initiate an aged mice experiment in January, which will be super exciting. But we really want to make sure that we develop the right experiment that meets a few criteria. Number one, it's scientifically valid, right? It actually predicts whether the compound will have a positive longevity impact in the mice. Number two is that it's relatively inexpensive. So I think a lot of the assays you discussed in terms of epigenetic clocks, looking at tissue pathology, looking at metabolomics, all of those are super interesting given infinite resources. I think we're trying to operate in a world where we get the highest signal per unit cost and time. And we're trying to figure out what is the fastest way to kind of kill bad compounds that don't work. And so we've been thinking a lot about these functional tests as well, these frailty indices, et cetera, cognitive. we think that, just like with Aura, how you can measure lifespan with computer vision, you know, setting up a camera and just tracking animals is much cheaper
Hmm.
than dissecting them, doing tissue pathology, and doing metabolomics and mass spec, right? So and on top of that, we're operating in crypto, which has the users are very much willing to fund good projects, but they have to be entertaining. And so it's yet another criteria that we have to solve
Yeah.
for is actually entertaining science. And so we think these functional tests actually are even more interesting because you can start to envision what we call the mouse Olympics. And so it's basically all of these different potential functional tests you can run on these aged mice to see and have a leading indicator of longevity effect. So, Matt, if you could design the mouse Olympics, what would be the different events?
Oh, wow. That's it. That's a really interesting question. So, uh, I think the real answer is I need some time to really think about it to do it well. But I just off the cuff, I think, like you said, cameras that just monitor daily activity, while maybe not super entertaining, I think would be really valuable because I think there's going to be both behavioral and physical changes that you can pick up on. I think in terms of function, there's what I would consider relatively easy, which would be things like rotarod for balance and coordination, treadmill, right, for physical ability to exercise capacity. Imaging, I would think about. This is a little bit harder to actually operationalize, but I would think about things like ultrasound for heart function. Um, but I think to do this justice, I think I need some time to really think it through. The comment about economics and scale is certainly valid. Of course, you always have to think about, how to get the biggest bang for the buck. Um, I really view the power of the invertebrates, the worms and potentially flies as an add-on, as the funnel, right? So that you're really only moving the highest priority, highest interest stuff into mice. So you don't have to test, hundreds of interventions. Then I think if you're smart about the way you prioritize, you might be able to get it down to a relatively small number of things you test in mice, maybe at a little bit higher resolution. The last piece as well, is how much do you prioritize health span or function, and how much do you prioritize lifespan? And there's no right or wrong answer. In some ways, it's almost a philosophical question in the field these days. My personal view is I want to know health span for sure, but I'm really interested in whether these interventions can robustly increase lifespan. And if that is something that you want to get out of the experiment, you have to make sure that whatever you're measuring isn't going to artificially impact longevity. So that's a design consideration as you're thinking about how you actually do these functional assessments. Obviously, terminal assessments like tissue pathology, you can only do, if you sacrifice the animals, you're giving up the longevity experiment. So that's something to pay attention to. And again, my personal view is, I'm just super interested in this question of, can we find anything that's better than caloric restriction? Because nobody has. I know we can, but I would want to know the lifespan effect at some point of whatever intervention we prioritize for moving forward into humans or companion animals.
Absolutely. Yeah, I think we're just trying to be as practical as possible about this. Of course, there's the whole philosophical argument of, living better versus living longer. We hope that some of these, we certainly don't want to advance something that may, increase longevity if it hurts health span,
Yeah.
right?
The
And
reality is
so,
it's obviously something everyone's concerned about. I have yet to see an example of anything that convincingly does that. So I'm not, I agree. We need to know, but I think if you slow biological aging, which you almost have to slow biological aging or partially reverse aspects of aging to robustly increase population lifespan. And if you do that, unless you uncover some other toxicity, you're going to improve health span. So I'm not convinced that's a real concern, as long as you don't have a toxic compound, which is unlikely to increase lifespan.
Totally makes sense. And then kind of along the same vein, beyond mouse experiments, of course, we all are looking to
Yeah.
humans, right? Which is ultimately the organism whose health and life we want to improve. So I guess the same kind of question applies, which is what is the cheapest, fastest, most scientifically robust readout that you would suggest that we run when we move to human kind of trials?
Yeah.
Um, with the caveat that, we're trying to, I mean, we're in the decentralized science space, right? Like we want to run decentralized trials on as many people as possible. The more accessible and easier the data is to capture, the, larger N will be able to service. I think that, when lots of biotech companies are thinking about designing trials, it's about how do we, enrich the data set as much as possible. But of course, that comes at a cost. We're not dealing with VCs here. We're dealing with individuals.
Yeah.
If you were to be dealing with individuals and kind of wanted to hit this equilibrium between breadth and depth in terms of readouts, like how would you think about this?
Yeah, great question. I don't have a fantastic answer for you. I think number one, we all wish that we could trust the epigenetic tests or other biological aging clocks to be the readout. I don't. And if for people who are interested in why I don't, check out the OptiSpan podcast episode on the biological aging tests. Right now, unfortunately, the direct-to-consumer biological aging test market is troubled. I'll just leave it at that. I don't think you can get any signal from the noise right now using direct-to-consumer biological aging tests. So regardless of what you think about epigenetic clocks and their fidelity as a readout for biological aging, right now, the way they're being marketed, there's just too much noise. So I don't think we can use biological aging clocks yet. Hopefully we'll get there. So in the absence of that, what are the kinds of things that we can look at? There's no right answer. I think we just don't know enough. The things I would think about, I really like oral health and periodontal disease and gingival inflammation as a readout of biological aging. Again, the reasons why that's in my head come from work that we did in my lab. Jonathan Ahn at University of Washington is doing work now in humans on this as well. But it's super non-invasive. Any dentist can give you a pretty quantitative readout of gingival inflammation when you go in for your dental exam. And there are more sophisticated things you can do, but it's really easy to measure. So that's one of the reasons why I like it. It seems to be pretty responsive in a short time frame, at least to some things like rapamycin. So that's one place I'd think about. Blood biomarkers of inflammation. And I think there's a lot of interesting work being done. For example, work out of the Buck Institute looking at inflammatory biomarkers with age, and Stanford as well. But even some of the more common, clinical chemistry markers of inflammation seem to track, I think, pretty well with biological aging. So, sort of basic blood work or more comprehensive blood work, blood panels, I think can be a pretty good readout of response to interventions. I like body composition and muscle function. Again, I don't know how sensitive those are to biological aging interventions in humans yet. We would certainly expect that if you're having a significant impact on biological aging over a period of a few years, you might see positive changes in body composition and associated markers of inflammation, perhaps being derived from visceral adipose. So those are the kinds of things that I would think about. I really liked, I don't know how many people listening are familiar with Joan Manick's work when she was at Restore Bio and before that at Novartis, looking at vaccine response in the elderly. I really like that as another potential readout of biological aging. We know that inflammaging, chronic inflammation with age is a major hallmark of aging. Most of the interventions that we currently understand to affect biological aging have a positive impact on immune function. Vaccine response in elderly people seems to be a pretty reasonable place to look for a readout of immune function. So I think about that. The last thing I'll say, though, and this is partly why I say we don't really know the answer, is all of these things that I'm talking about because we have reason to believe that the things we already know about have a positive impact, like rapamycin, like healthy diet, right? Like exercise. So it's a little bit of a circular argument saying we should measure the things that we know are responsive to the things that we think affect biological aging to assess biological aging. It's kind of the best we can do right now. But this is partly why I do favor functional measures, because again, when you think about health span, what we really care about is how well we're functioning. I don't give a shit what my epigenetic marks say if I'm functioning really well. So I think that's where I would really prioritize trying to identify what are the things we measure and are they really functional measures of health.
Okay, super, super comprehensive. Really appreciate that. So if you don't mind me kind of playing this back to you, so the biological aging clocks are borderline trash. Periodontal disease is a very interesting biomarker. So are the blood inflammation biomarkers. Immune system function, so how you respond to challenges from viruses, is a strong indicator, as well as body composition. And you've kind of alluded to this by saying, functional measures, but you didn't outright say it. So I'm very curious why not. VO2 max.
Yeah.
So we find this to be a super interesting biomarker just in terms of its signal to cost
Yeah.
ratio. Anyone can kind of afford one of these wearables. You can wear it with you all the time rather than it being this kind of fixed moment in time when you draw blood and you get a snapshot. It's this continuous marker. And so from a cost and continuous data perspective and signal, we think it's really interesting, but you didn't mention it. So any reason why
Yeah,
not?
great question. I think probably one of the things I would wonder about with VO2 max is how responsive that's going to be to let's just say a small molecule biological age modifier, right? I just don't know. So I think with things like immune function or vaccine response, from a molecular mechanism perspective, it's really easy for me to envision how 8 weeks of treatment with a small molecule geroprotector would cause a positive increase in vaccine response. It's a little bit harder for me to have confidence that VO2 max, in the absence of training for VO2 max, would show that same signal. Now, if you combined, a training regimen with a geroprotector, would you expect to see a better improvement in VO2 max? Yeah, probably so. So I could be wrong. It's just that I don't have a strong enough understanding of, exactly how biological aging mechanisms per se are impacting VO2 max as a biological readout. And it's super complicated by the lifestyle component, right? Whether or not people are active, exercising. If they're exercising, are they specifically training for VO2 max, right? So that's probably why that didn't pop into the top of my head as, a biomarker. I think as a discovery biomarker, it makes a ton of sense. And I agree with everything you said about it being easy to track, relatively inexpensive. I'd put sleep quality in sort of that same bucket, right? Really easy to track these days with wearables. I'd also put continuous glucose monitoring in that same bucket as a readout of metabolic health. So I certainly could see the value of collecting that data set and getting a better understanding of whether those things are going to be responsive to a small molecule geroprotector in a quantitative and robust way.
Awesome. But just to kind of get into it, if you could test any of the two markers that you said, and would it be, like body comp and VO2 max? Would it be, I mean, let's bucket wearables data into one kind of data point because that just requires, them
Yeah.
wearing
Yeah.
practically just them wearing a wearable.
Yeah.
So would it be wearable plus body comp?
You know what? I would actually turn it around and start more from the perspective of what are you willing to spend? And then think about what you can do, right? So, I mean, as an example, in our clinical program, I could say if we were thinking we would be willing to spend $5,000 per person to test an intervention. I know that may seem like a high number, but I'm just throwing it out there. There's
Yep.
a lot we could do, right? If you're only willing to spend $500 per person, that limits what you can do, and you just look over a 6-month period. So that's the way I would think about it.
Let's assume $500 6-month period.
So for that, you could do a fairly basic blood panel, a DEXA, and wearables, probably.
Great. Awesome. We'll write into the protocol. Awesome. Thank you so much. And sorry to, hard press you on these.
No, that's okay. Hey, this kind of brainstorming is important and valuable. So happy to add my 2 cents.
No, I really appreciate it. Just because this is totally on the horizon for us. And people are always asking us about the end game. Where we launch on the WormBot, but then where does it go from there? And so these
offline, offline, I'm happy to give you guys ideas on the blood-based biomarkers as well, because I can look at our sort of comprehensive set and pick and choose what you could do for a specific price point. So don't hesitate to reach out.
Fantastic. And then I guess like maybe a more general question. And I think you kind of alluded to this with the exercise and the diet things, but I know you're working on this at OptiSpan. You're probably consulting for a ton of, different people who are all interested in, these questions of how to live a better life. But, if you could kind of summarize, I know it's hard, but if you could summarize, important lessons you've learned that you would recommend people follow, just kind of like rules to live by, anything you can share on that front.
Sure. Yeah, I would say from a general perspective, where I tend to start is first of all, so we think about it in terms of 4 pillars of health. I know everybody's got, anywhere between 4 and 12, but 4 is easy for me to remember. We use active words: eat, move, sleep, connect. It's mostly around lifestyle, but eat is nutrition, move is exercise, sleep is sleep quality, and connect is both personal connection, mindfulness, as well as human relationships. So at that level, I think everybody pretty easily can sit down, spend 15 minutes thinking about those things in your own life, figure out where the lowest hanging fruit is, and focus on that to start with, right? And again, we've got podcast episodes on this. If you're interested, OptiSpan podcast, more information there, right? But I think that's an easy place to start. And then the other piece of information that I tend to use in my own life is whenever I'm thinking about trying something or doing something for my own personal health, I kind of ask myself, where does it fall in terms of likelihood of efficacy, or how much of an impact will pulling that lever have? There are things that we know work. I put the pillars of health there. They work. If you actually lean in, you will see positive benefits for your health if you focus on the pillars. There are some medications that we know work. If you've got diabetes, metformin works really well, right? If you need to bring your lipids down, sometimes statins, sometimes PCSK9 inhibitors work really, really well. So those really move the levers. Hormones, that's a big thing for a lot of people in their 50s and 60s. Get your hormones right. It's going to work, okay? Then there's stuff that falls in the middle ground, might work. I actually would put rapamycin in that. It works for some people, I'm sure of that, but I don't know that it's going to broadly increase lifespan or health span for most people. Some supplements I'd put in that bucket. And then there are things that if they work, they're incremental. I don't spend a lot of time there. So I know a lot of people who spend all their time in the incremental, might work, might do nothing category. So my advice would be, don't spend a lot of your time in the incremental category unless you've really dialed in the stuff that works. I think those are kind of 2 places where I would start as big picture. What can people do, and where a lot of people maybe are inefficient in their approach to improving their health.
Awesome. Super helpful. Really appreciate all of that. And then I think kind of enough with the serious questions. We've got a more fun question that came from the community, which is there any evidence for nicotine's impact on longevity?
Oh gosh. I hesitate to say no to any question that starts with any evidence for because you can always find something in the literature to back whatever point of view you want to take. I guess I would say I think there certainly is. You can come up with biochemically, biologically plausible ways that nicotine could impact at least health span. Longevity, if there are longevity experiments out there, I don't think there are many, or they are robust. So I would kind of put lifespan probably not a lot of evidence, but it doesn't mean that there couldn't be an effect there. But certainly it could have effects on health span, at least for some people. I think the question always is, what's the risk benefit look like? And most of the ways of delivering nicotine we know aren't healthy. And certainly there is going to be risk of side effects from nicotine. So I don't think I definitely wouldn't put that in my bucket of things that I'm super interested in right now. Sorry, I know
Fair
that was
enough.
sort of a dodging the question answer, but I don't have a great answer, and I don't feel really
You're all
strongly
good.
about it.
You're
It's sort of
all
like one of
good.
these things when people ask me questions, I'm like, meh, I don't care.
Yeah.
Sorry for that.
If it's not gonna move the needle like crazy, then it's not really worth focusing
Or
on. I
or or
totally
maybe
get
it's
it.
just a we don't know, but there's enough reasons to be worried about it that it's not something I think is likely to be a big thing down the road.
Yeah, that's super fair. I guess one thing I'm super curious to hear your thoughts on is there's so many personalities and voices within
Hmm.
this space. Like the longevity science space is becoming much more a religion than a science, I would
Oh
say,
God.
just, having followed it for about
Yeah,
a decade or so.
yeah.
There are certainly the preachers who seem like they have good intentions. It's very difficult to kind of piece apart, who's seeking truth, who is just trying to pump their bags. I guess would love to hear, I'm not asking you to kind of like downplay anyone's credibility, but maybe who are some of your favorite people and sources of truth in the longevity space?
Yeah, I mean, it's a great question. It's a tough one for me to answer because I obviously don't want to call people out in a negative way, although I have in the past. So, but I guess I would say first of all, my training, of course, is as a scientist, so I tend to gravitate towards people who are scientific minded, and I totally get what you're saying about science versus religion. I think one way you can tell the prophets, and some people call them futurists, from the people who are really science focused is you know um, you know faith is belief in the absence of data or evidence. And so if you don't have really strong evidence for something that you're claiming, that falls, in my view, in the realm of faith or religion. So that's kind of where I draw the dividing line: is there actually good data to support the claims that people are making? Another rule of thumb that I think is pretty sound is if somebody lies to you once, they're probably going to lie to you again. You probably shouldn't be getting your information from those people. So that's kind of how I, make my own decisions about who I listen to and who I trust. You're right. Sometimes it's hard to tell intention from, just being confused or uninformed. Some people have really good intentions and they spread misinformation just from a lack of background knowledge. But there are some people who clearly, like, it's crystal clear, they have lied about their own science or about other stuff. I wouldn't trust those people. Okay. Having said that, who do I really like trust? I'm not big, honestly, in the influencer space. Like, believe it or not, I very rarely watch YouTube videos on longevity. I don't really read books, popular press books on longevity, mostly because I see stuff that's wrong and it just frustrates me. So it's not worth my personal time to get frustrated about stuff like that. So I'm not a big consumer, although I am being more of a producer these days. I will say, like, in terms of influencers who I do follow regularly, I do pretty regularly watch and listen to Peter Attia's content. I think he's one of the more responsible voices out there. I think, sometimes I disagree with what Peter says. Every once in a while, he'll say something that I don't think is completely correct, but he does his homework and he tries really hard to put accurate information out there. And I've learned a ton by following his content. So I feel completely confident, recommending his stuff as one of the more authoritative sources out there in the longevity space that I think has a pretty reasoned approach, and a pretty sound voice, meaning it's not too far on the hype side, not too conservative or pessimistic. So I would put Peter up as kind of the gold standard, in my view, of really high quality content in the space these days.
No, and not only are you consuming Peter's content, but you're also producing it. I think you've been on multiple of his podcasts, and both are great, if I remember right. Um, but yeah, no, he's certainly been an inspiration to us as well. I think maybe where we kind of deviate from, Peter's philosophy is, he's very much a health span person. He's not really thinking so much of how do we radically
Yeah.
extend lifespan, but rather, how do we optimize with the tools we have. So, very much have been inspired by him.
And let me just add, so I don't disagree with you. I think I tend to be a little bit more on, we need to do both, right? We should absolutely optimize health span, and we should be going for longevity. And I haven't actually talked to Peter about it yet, but I've mentioned it a few times that, he's all like medicine 3.0, right? Which is the shift from reactive disease care to more sort of proactive healthcare to optimize health span. And I'm like, let's just skip over medicine 3.0, go right to medicine 4.0, which is medicine 3.0 with geroscience and artificial intelligence. And if you want to get to medicine 5.0, which is really moving the needle on longevity, that's where Aura Biomedical comes in and all the stuff we've been talking about today. So there is a path. What we really need is the will and the resources to take that path.